Yeah, as you guessed from the title, I'm not involved in DUP15Q, but I want to bring you some experience we had with another disease due to a duplication. Several years ago, we started to work on this disease, which is an Autosomal Dominant Leukodystrophy. It's a form of progressive neurodegenerative disease with an onset between 35 and 40 years. The patients, when start to have symptoms, have a quick progression and more or less between 5 and 10 years of life. In 2006, it was found that the gene responsible for this disease was found, and it's LMNBI. And the mutation here is always a duplication of the gene. So in patients, you have 3 copies of the LMNBI. And our idea to move towards a therapy was to try to find a way to switch off at least one of the alleles of LMNBI. To do this, we thought about RNA interference, which is widely used in the laboratories, but it's now also used in therapeutics. There are several even trials in several diseases using RNA interference, and you can use a 22 double-strand RNA to switch off the gene of interest. In the lab is very easy. It's quite a little bit more tricky to move into therapy. We were worried actually about one thing, that when you have a patient, you have 3 copies of the gene expressed in the high expression of both messenger and protein of LMNBI. But if you use siRNA, an interfering RNA, what you can get is an excessive reduction of the product, and 2-liter products can be deleterious and could have been deleterious for patients even more than the duplication. There is a knockout mice with a neurodegenerative phenotype. So we thought to move into a different strategy, trying to switch off just one of the 3 alleles using what it's called an allele-specific silencing. Allele-specific silencing is already used in other disease genes when you have a dominant negative mutation or a gain-of-function mutation. For instance, in spinocerebellar ataxias, it has been proposed. And basically, you have to find a SNP in your gene, which is expressed in a messenger RNA, and you have to design a specific interfering RNA which is able to switch off only the allele you want to switch off. What you should get is a downregulation of your gene down to the more or less the same amount of product that you get in controls. So we started designing and purchasing a library of siRNAs with what it's called a 19+2 geometry. We designed 19 siRNAs that were tested using a dual reporter system, which is shown here. It's a psi-check plasmid which is transfected into ACT293 cells. You have a luciferase reporter, which is together with a renilla reporter. The renilla gene is fused with a C-term with a 3' UTR of LMNBI, which contains the region target for the siRNA you are going to use. So testing all these siRNAs, we got the results you see in these slides. From the left to the right, you see the results for the siRNA we used, and the green and the orange bars are respectively for the matched siRNAs and for the unmatched. And the 4 siRNAs boxed in blue are those with the best results. We used 2 different concentrations. and you want to have a orange bar near to 100% and a reduced green bar, which means that you have a specificity for your sirna, which downregulates only the allele you want to downregulate. So analyzing LMNBI gene messenger RNA in ADLD fibroblasts, we showed that actually the messenger was downregulated using an allele-specific sirna. So that was the, we used also a test to see the two alleles, and you see here the two best results in which the duplicated allele in this case was fully represented, and the non-duplicated allele was shut off. In this case, you get a completely reduction to a normal level of LMNBI. We see that also the protein is reduced, and it's reduced to normal levels compared to samples in which the sirna was not used. And then we could test two features of the cells from patients. These are fibroblasts from ADLD patients. They have nuclear invaginations and nuclear blebs, and there are transcriptome alterations we could test after the use of sirnas. Concerning nuclear abnormalities, you see that the patients' cell lines have blebs in almost half of the cells, while in contrast there are blebs, but they are only around 10%. If you treat the cells with sirnas, you get a normalization of the cells with around the same amount of abnormal nuclei as in the contours. And also using transcriptome alterations as readouts, we showed that we showed actually previously that the two genes were altered in ADLD fibroblasts, LRRC15, which is completely restored to normal levels after treatment, and this gene, which is REVER2, which is reduced but not completely restored. Probably that's a problem of time treatment. We need to treat cells for a longer time in vitro. So that's all. Actually, our results seem to show that using using a SNP, which is with a high frequency of heterozygosity, you can get a specific an allele-specific silencing of your gene, and that this approach can be used in case of gene duplication such as this one. Just the last slide, just to thank people who worked in the work, and especially Dr. Elisa Giorgio, who has done all the work, and who is a talented postdoc in my lab. Thank you.